down from winter cold Central Europe. Well, don't be afraid, it's a long title, but the story is rather simple, and I promise you will not hear about any genes or any specific sequences, so I hope that you still keep, I know that the last speaker is always a problem. Okay, I try. Well, I step back a bit. I by purpose put the Mendel because I'm sure that even so you come from different continents, I'm sure that all of you have heard about P because of this seminal work. Actually, as you notice, it's precisely 150 years when he had a talk, so it was happening 8th of February, 8th of March. And I hope to come to that on the very last minute of my talk. Actually, he was very wise to choose a P, as I think most of you realize. He would have a trouble to choose something else which he was testing. I step a bit, well, you heard already yesterday and today several talks that there is a diversity problem in terms of the domestication, and since domestication is my key interest, I just would like to show you some specific numbers, how it looks in terms of legumes. So here is a chart which might not be exactly accurate, but I think it reflects very nicely the scenario. So these are percentages, but here are the specific numbers. So we do have about 1 million accessions worldwide of these particular legumes. And I highlighted P and also highlighted lupin by purpose. So usually what we are definitely lacking is a wild relatives, because simply the germplasm has been over the last, let's say, decades constructed mostly focusing on collecting the land races or breeding materials or varieties. There are situations different. Of course, lupin, like being successful crop nowadays in Australia, is relatively recent. There's 150 years of basically domestication, rather rapid. And of course, there is something different also in forage legumes, which you have a large proportion basically of the wild species kept in a germplasm. Specifically, if we stay with P, I was involved in some kind of inventory. I think it reflects more or less the situation in most of the other legumes. So we do have about, let's say, 90,000 Ps worldwide. But of that, we have basically about 2% only, which are wild, really wild accessions. And only a few of them we know sufficient collection site data, so to say passport data, which are definitely relevant, I think, which also Doug Cook has touched upon in a Sicer story. So this is just to illustrate how the situation looks like in P. As I said, it's very, very similar to the situation in the other crops. So, these, let's say, green dots in this case, well, they represent not just only the cultivated, but basically mostly cultivated and primitive domesticates, if you like. And this colored one, as I come later on, they represent the diversity being presented in these few accessions, as I stressed, so basically few hundreds, which comes from the wild material. Situation is quite clear. Well, the P it's a bit neglected. It's not the mandate crop in any of our, let's say, the CGR centers. And essentially, it has been quite well investigated in Europe. And also, unfortunately, is neglected a bit economically, except of Canada currently. So this is a situation, how it looks about this number of accessions being surveyed in a European germplasm collections. And these are basically the different core size. And what is definitely, I mean, just different representation. So here is a diversity of the wild ones, which are definitely recognized by any algorithm which you like, by any criteria as the most diverse. So nothing surprising about that. So this is just to give you a flavor how far we can go. So this is a domestic gene pool. There is something interesting happening here, something which is basically what we call Central Asia, so Nepal, Afghan types. And here is, I would like to focus your attention to this branch, which is, I don't know if you can read it, is a Pisum fulvum. So that's what I will be talking about. So this is basically as far as we can go. This is a kind of phylogenetic representation, as you like. And this is essentially progenitor of the cultivated P. So we do deal about with 2, maximum maybe 3 species in P, so it's not that much. And this is the, let's say, graphical representation just to illustrate you where the wild accessions come from. So the center of diversity is definitely the plantar crescent. It's pea is one of the founder crops, along with lentil, chickpea, and other cereals. But it goes well. You can still find some wild peas in Morocco and in the Mediterranean, and even so, the Mediterranean, of course, has been heavily influenced by the human activities. Particularly, it's interesting is also Caucasus, and you might go a bit in Iran and Caspian Sea coast. So that's how, so these yellow, I don't go into really details, but these yellow ones are Pisum fulvum, which is quite specifically around the Israel, Syria, and a bit of southern Turkey. And as the title of my talk says, well, I try to explain you why I have chosen to use Pisum fulvum. So this is another representation what you can use for in working with pea, how far you can go with a classical hybridization. So this is a classical Harlan concept of the gene pools. So there are different concepts like taxonomy pools and so on. We cannot go too far. So this is, let's say, the cultivated and the progenitor, if you like. There is some Ethiopian pea, which is quite interested. And as I showed already, the Pisum fulvum is where we can go without employing any biotechnology tools. So this is, I think it's obvious, I don't need to stress it more. Definitely the domestication and further modern, basically breeding from the land races has put some bottlenecks, and so we want to do something about that. We want to get a bit wider again, and at least to recover something from the wild species. So there are several ways to do that. I think you heard already today about the magic populations and some other approaches. And I decided some years ago, basically as a side project, so not to do the specific crosses as it was shown. So we know that in wild species you might expect some resistances. Like as it is shown here, there is a Bruchus resistance identified in a Pisum fulvum which has been deployed in Australian. And also there are some reports about the resistances to specifically here polydyl mildew, but you can imagine there are much more. So I didn't want to do this kind of classical biparental crosses, because of course also breeder doesn't like come back to the wild because that's have tested since ages, but they always come back because these wild materials always usually dominant. So I was very much inspired by work done by Dani Zamir on tomato about, let's say, pioneer about 30 years ago. Now it's widely used also in rice and other crops. So about 3-4 years ago, I decided to choose, let's say, 2 different parents. So Pisum fulvum, as I said, the farthest as you can go, and basically the progenitor of the cultivated pea. You might ask why particularly these 2 genotypes. Well, there was some purpose, but a lot of subjective selection, because of course at that time we didn't have enough information, and we are still not having enough information. Well, those parents are being tested currently, as I will show on the end. They are basically, it's now in the European project, so they have been tested for the resistances and so on, so we will have more information on that. And the concept, as I said, I promise, the story is quite short or quite simple. So it's nothing spectacular, so it's just classical genetic. You cross, of course, but then you do several backrosses, and what you want to have on the variant, in the ideal situation, is like a series of lines. So this is one line, it's like this, so it's a chromosome and the line. And you would have, in ideal situation, like part of the chromosome being substituted, so that's why it's called chromosome substitution line, or introgression lines, if you like, being substituted with a wild donor. So that's why it takes quite a while to do that. Just to, let's say, illustrate that the pea, maybe that's the reason why it has been abundant for genetical studies, because the genome is pretty large. It's about 1 giga more than a human. And it's quite full of the retrotransposon, not surprisingly. As I will show you on the end, there is an effort, of course, to do a pea genome. I can just say that the only one chromosome could be flow sorted, so let's say by size. So this is only one which can help, as it was pointed out today, in, let's say, 3TC genomes and so on. So there is a bit problem with doing this. So this is a principle. Again, it's just to remind you, okay, here we have 50, 75, and 97, and so on. So you want basically do the recurrent, the back parent. So this is a cultivated material, and this was our wild donor. At that time, as I said, it was a side project at that time. It's one of my focus now. So I decided, because I'm dealing with hundreds of lines, and I just wanted to monitor, the situation and to choose which line to follow up and wait for the, let's say, the better marker systems, which I will come in a minute. So at that time I decided just to do, let's say, to choose the gene base or microsatellites even, which I know precisely where there are. Of course, they are relative, so to say, here are big gaps and so on. But it was reasonable, let's say, decent. So there are varying in the spacing. We do have much better maps nowadays, but this is a situation where I started about 4 years ago. And I think it was still okay. I'm quite happy with that. So this is just the snapshot of how some of the lines, as I said, I didn't tell you about the cultivated parent. So the wild parent, as I said, was one particular genotype. The cultivated parent, in this case, it's a classical commercial variety, the semi-leafless, or aphylla, if you like, the Czech Terno variety, which is quite successful, good yielding, and it's grown not just in Czech, it's quite useful. So that's what I have chosen. And as you can see, there have been 3 backrosses done, and then 4 selfing. So this situation from about the last year to, well, yeah, since then there are 2 more generations now. This is a graphical representation, so this is a relative. Of course, the distances can be a bit, let's say, different because you can put the spacing here. But it gives you some flavor. So there is still heterozygosity, as you might expect. And as you can see, it's quite interesting because I expected first quite large blocks to be held by let's say, this wild parent. But in fact, it was quite broken. There are several, well, we can maybe leave it for a discussion if I have time, why it could look like this. So as I said, the ideal situation, which we are going to, but of course it's unlikely to happen, because if you are familiar with introgression lines, even which has been really done in tomato a long time ago, or a rice, they never end up with just single fragment. There are usually something somewhere. So we included these parents in several genotyping platforms, including SNP, Illumina, including DartSec, and others. So the parents are pretty well genotyped and characterized now, and basically now will be the choice which to choose for much denser genotyping once they are sufficiently homozygous. Well, there are some theoretically interesting things happening as well. Since I'm based now at the university, so I can play a bit more things. So I didn't tell you, but initially this process has been made in reciprocal manner. And what was interesting since the very beginning, there have been some pale or even albino-looking plants, and we have been wondering what's going on. We suspected, because there are some colleagues in Russia, in Novosibirsk Academy, we just published about this nuclear-cytoplasmic conflict, and I can tell you there are already 2 genes identified, so nuclear and chloroplast one. So the story is going to be really interesting. And we noticed that even when we do it reciprocally, not surprising, I mean, there is report about the biparental inheritance of chloroplasts. So the mitochondria behave very well. It goes from mother, as you might expect. But the chloroplast will somehow get eliminated from fulvum, whatever you did. And we tried actually several accessions. This makes a problem, of course. You are losing these lines because they are not very viable. And it makes a problem, including the alatius, which I'm not going to share with you because it's in early stages of development. So this was Sterno. So basically the situation, how it looks about a year ago was, as you might expect, well, there is this part of the heterozygosity still. There is a bit less of the cultivated or recurrent parent, which you might theoretically expect. You might speculate why. We know that there are, I didn't mention, there are karyotype differences. So there is some segregation distortion, definitely, because it influence initially the fertility of the very first 2 generations in general. Then you approve. So as I said, there was pretty decent number of markers, quite few, but as I said, I think it's quite okay for the purpose we have made it since beginning. And also it was a side project. I had to make some kind of course, some report on the end. So this is keeping in mind that these lines are still not fixed, but it's interesting to record some, and we look at about 14 different quite easy scoring traits, like number of nodes, the height, which is quite obvious. So there are quite some dwarfs, and some are as tall as I am, or even higher. But it's interesting, it's arranged in this way, and all the others, the charts or blocks. So here are the parents. I don't think you can see it very well, but here is one of these parents. This is a fulvum, and this is another parent from another cross. Interestingly, the number of pods doesn't matter if this plant has like nearly 2 meters, or if it has such small little dwarf. So this small little dwarf has, of course, a small number of nodes, but it's quite fruitful. I mean, it looks like a small tree full of these pods. So yeah, this is the idea behind, which I should say on the very beginning, is of course to have something permanent. So not to make just simple cross, but basically to provide the lines very much like recombinant inbred lines, but with this single, or ideally single fragments introgress, which could be used for various purposes. I didn't target any specific trait, as I said, I am not targeting for any resistance specifically, but they should be there because we know these parents are interesting in this aspect. As I said, I'm interested in domestication and particularly in legumes. So that just to give you why I said in a title that it could be used for potentially for the gene identification. So this is one trait of interest is pod dehiscence. So as basically all of the domesticated legumes, similar to cereals, they don't dehisce or split the pods, or they split it relatively with a kind of mechanical force, while the wild ones, naturally, of course, they dehisce quite readily and spread the seeds. We know that where it is, the locus has been located since a while, and it's quite nicely corresponding basically to the region, because once you score these phenotype, well, you see that these are lines, and there are much more of that. Also, we have been losing some of these lines due to these infertility issues and so, but there are some which can be, we know that this fragment is basically somewhere there is a gene responsible for that. As I said, I promised that the story is rather simple and it's still ongoing. Also, I prefer some other to talk, but Rajiv decided let's talk about this one. So this is a part of the, fortunately, the project which is started a year ago, but it continues for 3 more or 4 more years. And there are partners, as I show you on the end, which will do, or which will help me with definitely much higher density of genotyping. So, as I said, there are 2 principal populations being established in various stages of development. And yeah, they're aimed to be really basically for anyone which is interested in P. So these are, okay, these person helped me a lot in the beginning. These are partners which do, we do have 13,000 SNPs mapped on the parents, and they can be readily already deployed in some of the lines which are, let's say, sufficiently homozygous. And these 2 partners also within this Legato project are helping with kind of medium genotyping of these hundreds of lines. Okay, I still have good time. I promise that I will not kill you with too much data. I think it's the story. I just wanted to show you that I think introgression lines are quite good too. You might argue, okay, which parents to choose because you spend some time establishing these lines. That's definitely true. But I think once you have them, they are definitely quite useful. It's a different approach from magic population or nested mapping. It's a different strategy. But I think if you look at really tomato story, there are good examples that you can dissect in this system with these tools. So I use the rest of my talk, well, just 3 more slides, don't worry, it's not going to be much. So I mentioned already Mendel in the beginning. So since I work in P and being from Czech, I think it's quite obligation to touch this. So not argue, of course, many things happen in genetics. The rules are about the same, but we shifted to genomics, and I mean nowadays it's so dense that you can put every month happening something significant, either methodologically or let's say as a discovery. So I would like to, well, unfortunately I was given by organizers the leaflets too late. So you have been given quite late, I mean the old reprint from the last summer. Which was done quite quickly by Rajiv. Unfortunately, we didn't have this final one. So we want to celebrate these 150 years. I think it's appropriate to do so. And so there will be in very authentic place the conference, which is going to be open, or it is open. It's I give some 50 reprints in a registration deck. So. But you will get, I think you will get by email from organizers. So we want to basically touch what Mendel was dealing with. So he was not just doing P, but he was, he was trying some Hieracium. He failed because he didn't know about the apomixis. Then he tried some other aspects, and we know today that there was some epigenetics, so he would have a hard time. He tried something on Phaseolus, which would be also quite difficult because it was a quantitative trait which you couldn't handle at that time statistics. So we want to touch this. We already have some invited or keynote speakers. So you might recognize some names. And this is, let's say, the committee which I was proud to, let's say, put together. It's slightly biased towards the legumes, but I think it doesn't matter because each of us, of course, does some different things, not just legumes. So that's what I would like to invite you, and I think Rajiv will distribute it to you. So just the last thing. Well, as I said, the P has been neglected, but I think there is a legacy, maybe because of Mendel. So there is things going on the P genome, even so, despite that it's such large and complex. So there is a consortium quite loosely, but it's going well. And as far as I know, well, there are some possibilities that maybe the genome could be on the, let's say, end of this year or beginning of the next year, done by really proper, so to say, back by back. It's not just a shotgun story. Let's see how it handles with these retrotransposon things, but there are people involved in that. So, okay, thanks a lot. There is plenty of time if you like to ask. Thanks.
Okay, thank you. Thank you for finishing on time. We have good time for questions. Are there any?
Thanks for your really interesting presentation.
Oh, yeah, sorry.
I'm part of the P-sequencing Consortium, and just to clarify, there are some backs being sequenced, but as far as I know, it's not a back-by-back approach, which is being taken. It's a shotgun.
Well, I know it's a mixture. It's a different because, as I said, one chromosome could be sorted out, which is French, and yeah.
And that was your colleague Yaroslav, presumably.
Yeah, exactly.
Been working with that. Now, I was interested in your introgression lines, and they could be quite a useful tool to help validate the assembly. We're doing something similar in brassica, where we've got introgression lines of different chromosomes, and we're resequencing those to validate the assembly because it's not that easy to isolate single chromosomes. So is that material being used and analyzed within the P-genome project?
Possibly. Well, we might discuss it because there is a meeting next week because of this European and the French group is going there. So yeah, there is a possibility to use this, at least partly. There are some troubles because there are some translocations in the wild, or contrast to the cultivated material. But yeah, the reason why I've used, maybe I didn't stress it much, but the cultivated part is a camelor. So it's the genotype which is going to be the sequence one. So that was made by purpose. It's not so useful commercially anymore, but it's basically, it will be a kind of model. So that was done like this, in this mind, yeah.
we're actually sequencing isolated chromosomes that Yaroslav are producing for this consortium. I'd be interested if you're happy for us to do this to test some of your lines as well, just to see what it looks like.
But I have one basic question. Can you please give the difference between phenotypic difference between Pisum, fulvum, and what you have mentioned, sativum?
It's hard to hear you. Sorry for that.
I have one doubt. Can you please give the phenotypic difference between fulvum and sativum based on flower color or hilum color, curvature of the pods?
What do you mean? The difference between the
phenotypic difference.
Phylogenetic or?
Yeah, phylogenetic.
Well, these are distinct species. There is no doubt about that. And the phenotypic one.
Yeah, phenotypic, the flower color.
Well, I mean, depends what you look at. They come from very different habitats. Even so, they come to in touch. I mean, in Israel, in Syria, you can find them sympatrically. Well, the fulvum, it's quite like rocky growing, let's say branching quite low, and definitely, let's say drought tolerant, because it's really, it's like adapted to the really dry conditions. Well, depends where you look at it. So the distribution area is broad, so you find different habitats in a piece. So depends what you want. But they differ, yeah.
Thank you.
Okay, thank you.
I guess that brings us to the end of this session. Please join me in thanking all the speakers for the excellent presentations that they have made.